So, are you familiar with some drugs in your family on the blood pressure or painkiller? What does they do? How much of them do you take? 1 pill, 2 pills, 1 milligram, 2 milligram. This part is related to that. Yeah. Designing drugs, see, you know some of these drugs. What is the algorithm for the patient that they have gout? Or atrostatins or simvastatins. They are used in patients, they are in high cholesterol levels. And methotrexate as a counter-cancer drug. You are not supposed to learn these drugs. I just put them as an example. None of them. And captopril. Are you familiar with this one? Many families, they actually use different types of the anti-hypertension drugs. One of them is captopril. And Bicomarol is a vitamin carboxyl reductase. It's an anticoagulant. Or you might have heard the name of the warfarin, something like that. In different countries, they have different trade names. Warfarin. See, all of these drugs are targeting enzymes. So, in this part, we are going to talk about what factors can affect enzyme activity. 3 factors. One of them is cofactors and coenzymes. Vitamin deficiency is a kind of not having that enzyme. So vitamins are essential because of all of these enzymes are working with vitamins. Another one, the environmental conditions. pH and temperature. Both are essential. In today's lab, actually practical class, we are going to see the environmental effects on the enzyme activity. You might ask a question, why should I learn these things? Yeah? Why should I learn environmental effects? I'm not going to burn myself or put myself in a boiling water. Why should I actually learn the effect of the temperature? But here, when you have a fever, more than 42, if you reach a 45, you can't tolerate that, or no one can tolerate that. They will die because all of these proteins, many of these proteins will destroy in 45. And this is that they can't actually. And the other thing is that another conditional or environmental factor is a pH. They should have optimal pH for working. You might say again, why should I know? My body actually already, without thinking that and knowing that, my body also actually does the best things for the control the pH. Yeah, we know, we need to know this because we designed some experiments in the lab. If you go to the medical lab, if they are going to measure your enzymes, they should have optimized the reaction. Industries, they need to know that. And other things, enzyme inhibitor, as a doctor, you need to know that. Or as a pharmacist, they need to know this one because they want to design drugs. And they finally, they want to give you, let's say, 1 pill or 2 pill or 3 pill. How much of that? Or 1 gram of something, 1 milligram of something, or 10 milligram of something. What? How much of that? Because if you take more of that, it can be toxic. So, we will talk about the cofactors and coenzymes in a vitamin part. I'm skipping this part. Just go to the environment part. High temperature or different pHs or even substrate concentration. They can all of those things can affect enzyme activity. See, for the temperature, the increasing the temperature, see this is a 0 temperature and 10, 20. If this temperature, activity of enzyme is low, you are going to see in the lab. With the optimum temperature, 37, you will see the best activity of enzyme. Many of enzymes in blood, they work around the 37. Because of that, our temperature is controlled around the 37. And those enzymes are optimized for that temperature. About the pH. In our body, we have different parts of the body we have a different pHs. Like, best example, stomach. What is the pH? Around 2 to 3. So, we have a specific enzyme. What is that enzyme? Pepsin. So, pepsin is the enzyme works on an acidic pH. If you put this enzyme in alkaline pH, it doesn't work. And we have some other enzyme like human amylase. It works on neutral pH. Even more acidic. Around 7, less than 7. And also we have some other enzyme, trypsin. It works on an alkaline. These are examples. Mainly they work on a neutral pH, around the 7.3 to 7.4, around that works. But some others they work in a different pH. And we need to consider those things for designing the experiment in the lab or consuming best recommendation for taking specific drugs or treatments. So the third part is from that things that control the enzyme activity is the substrate. This substrate could be your actually taking everything like food or everything that we get, they are substrate. For glucokinase, when we get a glucose, what's that? That is a substrate for that. So sometimes it could be drug for that enzyme. It's a substrate for that. So for prescription of drugs, let's say how much of this drug should I take, or how much of this drug can inhibit. When you take these drugs, what's your goal? What is your action target? You inhibit this enzyme activity. Let's say I have an enzyme, and you are going to inhibit me. Before inhibiting me, you should know how I work. I work like this, or I work like this. Which one? Studying my behavior is called kinetic of enzyme. You study my speed. You should know my speed to control me. If I actually be so fast, you can inhibit me here. If I be so, let's say, lazy here, if you so fast something, it should not look like here. And I work also again. So, again, studying enzyme behavior or speed of enzyme is called kinetic. And kinetic is important for what? Because we should know the speed of or how to work of enzyme and then give drugs. Without that, we can't. So, there is a graph here. You can see in x-axis we have a concentration, and in y-axis we have a speed. Here, let's say we have a sum of me, for example 6 of me as an enzyme here. How much of substrate we have here? No substrate. See, this is 0. It says all of these enzymes are free. How is their speed? Because there isn't a substrate here. There isn't any product here, and the speed is low. Because we are just. We are not doing anything. Because we don't have substrate here to work on it. But with increasing the substrate, what happens here? Some of these enzymes start to work. Let's say half of them are busy, or 20% of them is busy and working. And with increasing the substrate, what happens? All of those enzymes are busy. All of them are working. And because they convert to everything, and if you add more substrate here, they actually work. They actually convert to something. When all of them are occupied, you will reach a maximum speed. Let's say 6 enzymes here converting everything. After that, if you increase the substrate here, you can actually increase my speed here. They will accumulated there. What is that? This is this code. See, you have all enzymes occupied. You have more substrate here, but your enzyme speed is the latter because let's say 6 of you are here and just convert here. So we are working in a high speed. no matter how much of the substrate are here, 1 room, 2 room, or 1 truck, or let's say, bunch of those things. This is the kinetic of enzyme. When you see the speed, this behavior is different from one enzyme to another enzyme. It's not like that glucokinase is the same to hexokinase. Let's say for the glucokinase and hexokinase. Some of these enzymes start working when 10 of molecules are here. For the hexokinase is like that. They start just working. If they see glucose are there, they start working. But for the glucokinase, they say, no, I don't like to start working. They wait, when they see a lot of those things. See differences between the hexokinases and glucokinases. Hexokinases, if see just one glucose start working that, but glucokinase waits a lot to reach a level of the glucose and then start working that. So you might say that what happens is that because most of them they are inside the cell, hexokinase is working, but glucokinase is there. Why we have glucokinase there? Because if this hexokinase works on a pathway for producing the energy, the other one is for storage that. When it goes up, it says, yes, it doesn't need that pathway. We have an extra of this, and it's going to keep those things. It's going to keep them as a glycogen. So enzymes are like this. So this different affinity. What is the affinity? Liking something. Hexokinase like that compound so fast and start working with minimum concentration of that, it reaches the high speed. But the other ones need more substrate to reach that maximum velocity or speed. So this is the equation. I will back to this one. So we have an equation here. What it says, how to calculate that? How to calculate the maximum velocity? Again here, see this substrate going up, and we will reach maximum velocity. In this area, no matter if you increase the substrate, you reach the maximum. So this point is a maximum, this is a velocity. This is the maximum velocity. Where is the half of that? Here. If you plot a graph on substrate part, there is a point. What it says, on this concentration, we have half of the speed. Let's say, in 2 molecules of the glucose, I have half of the speed, not maximum of that. What's the maximum? We know that. What's the half of that? He said 2 of that molecule or 4 of that molecule reaches my speed to half of the speed. So that concentration shows our affinity or liking something. On that concentration, I reach to half of the speed. So between the enzyme, this Km is different. And this Km is the best indicator for giving level of drugs. Because if you, let's say we have some substrate here. I'm supposed to work on them. And you know my speed and my liking that compound, you can put enough of other compounds can make me busy and stop my working, real working. Enzymes are like that. We give them these drugs, then make that enzyme busy to stop that actual pathway. So knowing this Km is a constant for every enzyme that is different. So let's say, for example, how many of, let's say, boxes should be here you start reaching your maximum velocity? You might say with 4 cartons or 4 actually boxes. How about the others? You might say, I will start when it is around the 6. I will reach my half of the speed. maximum speed. And it could be different between the enzymes, that one. And then I know that one, I can prescribe substrate level, like drugs, inhibitor. So I can put some other inhibitor beside you and say, this level of that compound, if I put more, what happens? It can make you busy, and you can do everything. Or it can be so toxic. It's not like that accumulating here. If I accumulate more of that here, they go everywhere. They might go the other enzyme, they inhibit that enzyme. Or other molecules. So because of that, we need to know that Km to prescribe exact concentration of something that is good for that pathway, not toxic for other enzymes or other pathways. So the formula is like this. Let's say this is the initial velocity, when it says when you want to calculate everything here, the concentration or maximum velocity, or this is a Michaelis-Menten, two scientists. Most of them are started with, one of them was, she was one of the best greatest in biochemistry, Menten, and another one, Michaelis, both scientists. They calculated this equation, and the name M comes from initial names of two scientists. It is a constant for every enzyme that is specific, and you will have this equation. One of your exam questions is going to be calculate. If you look at the question for this question, you will see I give you some of information about the maximum velocity or concentration or initial velocity, and you are supposed to calculate Km. But you know what is the Km? It's a kind of knowing how much of that compound are be liked by that enzyme. So usage of that for designing drugs mainly, or consuming. When you get more than enough carbohydrate, what happens? Your hexokinase starts working, but if you have more of that, you inhibit that one, and the other enzymes start working to accumulation that. If you consume more carbohydrate, what happens? All of those enzymes are occupied because they are in a maximum velocity. What happens to that glucose? They bind to everywhere, every molecule. One of those places they can actually bound, where is that? Yes, everywhere. They can make a toxicity to everything. One of them is kidney cells, bone capsule, base membrane. They have negative charges to produce albumin to go into urine. But if the glucose are actually bound to them, they will lose actually the negative charges, and albumin goes to the urine. See, we need to know the optimum or the highest velocity of this enzyme to consume optimum level. If we exceed that one, what happens? They will go everywhere. Some of those enzymes here, they can be inhibited that one, or they can bind to everywhere. So this formula is for that. This equation shows the enzyme reaction. We have enzyme plus substrate. It can make enzyme substrate as a transition state. It can go back again. In some cases, it doesn't go to previous product. It depends on how much of substrate that you have. If you have more substrate, the equation goes this way, forward. And when it goes to this stage, it kind of goes back. But sometimes these products can inhibit here. Because when you produce a lot of ATP, you are not going to consume carbohydrates as an energy. Let's say final product of this is a production of ATP. When you have extra ATP, can we save ATP? Do we have just a small amount of ATP we can save as a creatine phosphate? We can't actually save ATP. You can say that I have enough ATP. I'm going to run forever. You can run. You can get energy not from ATP. You can produce ATP at the moment, not saving that. But you can save lipids. You can say, yeah, I have saved enough lipids. I'm going to use that lipids for the ATP. That ATP, because of that, when you consume more than you need, what happens? Storage. Storage in the glycogen. In the next step, storage as a lipids. Obesity.
Salts?
Yeah. So why? What happens? Like this. When you have extra product, it inhibits this pathway. Like when you get a lot of energy, so it stops consuming that glucose. That glucose goes other way, like storage. I explained this once. And yeah, some other two scientists, they saw that progress from that two scientists, they actually made a reciprocal of that plot. They actually converted this equation to this one. They just reversed, yeah, this velocity, like this one subdivided by divided by v, and they changed the equation from this type of hyperbolics to linear one. For linear, converting to this one to linear one, you have to reverse the equation. The name, they actually took that name linear verb to other science. You might see this equation also. They are safe. Because they thought if you want to find a specific point here to make a specific plot on the x-axis, that is because it's not linear, it's a kind of complicated, and you might have some kind of mistakes on finding the exact point, but they decided to make it linear. They are safe. So you can actually find with everything that you have. Some of the slides, let's say you might have concentration of the substrate or initial velocity or Km of a specific enzyme. There is the things that I'm going to talk about, the inhibition. If you destroy the structure, that enzyme can't work. Like a cutter wants to cut from, let's say from here, but if this area is like this, it can't work. And it will not work. So, next time, is it good use ultra-pasteurized milk for production of cheese? Yeah, it could be. Why it could be? Because on this one, by chance, we had just a small amount of that proteins. They were like that. I saw in some tubes it worked so partially.
But for us, it's not properly.
Yeah, I saw, but we tried it several times. It didn't work. I increased the renin and protein, both of them. It didn't work. We put it there for several times, long time. Even when we put them in 80 degrees, you know what should happen in 80 degrees? It should show aggregation after minutes.
Yeah.
Because that shows that level of the temperature degrade the proteins. Like albumin, you boil eggs. How it happens? It's a kind of making solid of these albumin. The same thing should happen in 80 degrees. However, it's not casein working, but it's aggregation of proteins. So, I will look for another types of milk in the island. I'm not sure I will ever find that. But I will look for traditional types of the milks. And so if I find that, we are going to repeat that tomorrow. If I couldn't find that, Dr. Tanai's session is going to be the next session with you after we...
I saw it in the lab. There was a blue milk.
Blue?
The label, it was blue, which means it's normal. Normal milk.
In the lab?
Yeah, yeah. Right where they filled the call it the beakers.
So I asked actually our scientist, she said, I will use this one.
Yeah, she used that one, but there was another one. It was full and the label was blue.
I'm going to check it. I'm going to check it. If it works here, I will use it. So, and next go to the inhibitors. It's same to our subject that we discussed now. What are the inhibitors? Everything can can inhibit enzyme activity. We have two types of the inhibitors if we classify in big group. Some of them are reversible, some others irreversible. Irreversible, when bound to the enzyme, they destroy the enzyme. And the enzyme will not work. But in irreversible, you can do some other things and or wait for some hours and have your enzyme activity. Two drugs. There's two examples. When you consume or take one pill of ibuprofen or gelofin, you're familiar with these drugs. They are painkillers. They inhibit a specific enzyme, but that is reversible. They actually make that enzyme busy, doesn't let work to produce some pain, actually intermediate, stop that. We have another drug that is aspirin or salicylic acid. What's that? It acts at the same enzyme. Here, it's not competitive, it is irreversible. When it bonds to the enzyme, destroy enzyme.
So the enzyme not regenerate itself again.
Regenerate, but it takes time. 2 days or 3 days for that enzyme. But for the gelofin or ibuprofen, if you take that drug and go to hospital and ask for some experiment, they will ask you, if you ask for some bleeding test or coagulation test, they will ask you, what was your last actually drug you take? If you say it was in gelofin, they will ask, when was it that? If you say 8 hours before that, they will go for you. They will start your experiment because 8 hours is enough. Because our body actually gets rid of that drugs and that enzymes are free to work again so easy and natural. But about the aspirin, if you say, I'm taking aspirin, so they're going to say, go home after 2 days or 3 days back here. Because they destroy enzyme. So, this is the whole idea for the competitive and non-competitive of enzyme. And so, how they work. Just let's see how they work. We know for the competitive ones, in reversible ones, in reversible types, we have two types. Several types, but we are going to just study two types of them. One of them is competitive. They compete. There is a, like, let's say, there is a stick like here, and some substrate and some inhibitor, they want to compete on this shape. So it is a competitive. And another one is called non-competitive. They are not exactly sitting on here. They can occupy this area like what? A changing levels. It doesn't let this work properly. They want to occupy. They can stand here. Not necessarily actually exactly on the active side. They can actually bond to the other sides and make it busy or, occupy that. So it is called non-competitive. In competitive ones, they just occupy active side, but in non-competitive, it is also reversible, but they inactivate that enzyme. Like, let's say, if I do something like this, I work to my hand, this is my active side, and something bound to my actually neck and doesn't let my hand actually move or work properly. It is non-competitive. You have some equation here, for example, for the reversible inhibition, this is a competitive enzyme can bond to the substrate. That's shape actually be occupied with the substrate or occupied with inhibitor. I is for inhibitor. It depends on the concentration of this. If you use more of this, what happens? You will not have this pathway. You will inhibit all. It is like that how much of drugs that you get. If you get more of this, you completely inhibit this pathway. But sometimes you see, I'm not going to do that. If I just inhibit part of this, let's say you have small amount of something or effective amount of something, you say, I'm going to have this pathway also, but mainly I want to actually inhibit that. And this level of drugs is enough. And changes that you see, what happens in terms of the, let's say, I am an enzyme, and I'm going to be inhibited competitively. What happens to two factors here? My speed, my actually working process time, and my, affinity or liking that substrate. Two terms, Km and Pm. Km is a kind of affinity. See, we have changes here. This is, what is this? This is no inhibitor, these were ones. But what happens in competitive ones, let's say, both of them are same, two scientists, they actually show it in two different ways. But, see, what is this? This is normal. But what is this? Yeah, with inhibitors. What happens with inhibitors? Km goes up. What does mean Km goes up? Yeah, exactly, low affinity. Or you need add more of that, let's say, for computing this. I have a substrate, normal substrate, and 10 of me be here, and another inhibitor, just two inhibitors there. How can I actually compute with them? If number of them actually goes up, we can actually move them out. So because of that, Km goes up. It says more substrate you need to compute that one. So that's the term for this and this explanation for this one. The Km goes up. How about the speed? See, that is speed is same. Let's say I'm just doing something like this. This is my activity. If you put some other things like this here, I will do that. If this one is inhibitor, it doesn't change my speed there. It just changes my affinity to this substrate or this one. So Km goes up, speed is the same. The increasance of the inhibitor. The first slide for this part is the same drugs. I'm going to show you these drugs are competitive drugs. Statins, corticosteroids, anti-blood pressure drugs, they are working on competitive. The most famous one is Gelofen. Do you use that? I use a lot. I'm not recommending that, but yeah. Sometimes you have headaches, or some days that you haven't had enough sleep. Yeah. Unfortunately, I had some kind of headaches, and I get one of these Gelofens, and all the time I'm thinking about the enzyme that is being inhibited by that drug. It stops that. Yeah, for migraine, it works for me. And in the future, you will learn some other terms. Oh, another best example. If someone comes to you and you will be suspicious about some intoxication on their alcohol. You know, in some countries, some people actually prepare alcohol by themselves at home, but unfortunately, instead of ethanol, they produce what? Ethanol. Exactly. Or in some countries, there are some, conflict between smugglers, and they try to contaminate their actually alcohol, add ethanol to those things to get rid of some of those users, and they say that's not safe. Come to us. Ours is safe. So, if you're in a hospital, what is the best treatment for them? Even at home, if you realize using what? Ethanol. Giving them more ethanol.
Give them more
See?
alcohol.
Yeah, giving them more alcohol. Come here. I'm going to give you a treatment. What is the treatment? Why? You might say, why? The enzyme is safe. We have a safe enzyme for process. Both of it. have a ethanol, it's a also OH. So sorry for part of this thing. It's not bad. Yes. Alcohol dehydrogenase, we have a specific enzyme. Like, what is that? What kind of, it belongs to which group? Alcohol dehydrogenase? Oxidation dehydrogenase, group 1. Alcohol dehydrogenase. It doesn't make a difference between these two alcohol, it just works for sulfur. For this one, it produces formaldehyde. For this one, acetaldehyde. See? This one is more energetic compound. It has some toxicity, some headaches or kind of confusion, but it's not toxic compound. This one. And finally, the next step is converted to
formic acid.
and this one is converted to acetate. Exactly. What is the effect of this? Blindness. It's everything. It's a fixative compound. In the laboratory, when you get a tissue, you can put on a formaldehyde and fix it. So, now, you are going to make this enzyme easy to our kidney, get rid of that methanol, because we have methanol. We never let this methanol convert to this. We make this enzyme easy with, you increase concentration of this, and it's a competitive inhibitor. We have methanol in blood circulation, however, it has a toxicity to kidney cells, but it is better than blindness or dying from that. We are going to sacrifice our kidney cells because this one is also toxic for kidney cells. But when we make them busy, we let blood circulation to get rid out from that methanol by urine. So we are going to see examples of how can you use competitive inhibition in exactly kidneys. We have a lot of examples. You can actually, if you're interested, you can find. However, I introduced you some of these drugs. And yeah, for the non-competitive ones, our inhibitors, if you remember, in competitive ones, we have like this. Our enzyme substrate can be, this is a normal process, but in the competitive ones, we had inhibitor can bind to the enzyme. But in non-competitive, it also can bind to what? It's complex. So it is kind of complicated. So the action is different than the competitive ones because they can bind to some non-active site of enzyme. It doesn't matter someone on this shade or not. It can bind here. So when it's occupied or unoccupied. So what is the effect on velocity and Km? What is the effect on velocity? It doesn't change Km, but the speed decrease. That is enough. For reversible ones, that is enough. Let's go for, you can see the differences on this table. It is for you. When you are studying that, review that. This information will help you. How about the irreversible ones? How about irreversible ones? You know the aspirin, one of them. Mainly they are toxic. they can cause this in many situations if you consume them a lot, or if you target some critical enzymes. Some of toxic come from outside, or some of toxic defense. Actually, they target these enzymes irreversibly. Let's see some examples. They are very toxic most of the time. They mostly bind to the functional protein active side and block the side. What is this? The previous example. Aspirin, this is, we have an enzyme in the body that's called cyclooxygenase. This is the active site of that cyclooxygenase. We have a serine, there's amino acids there. And this aspirin carboxylic or acetyl groups of that bonds to that serine and makes it covalent bound, and it doesn't go anywhere to destroy that enzyme. It's cyclooxygenase. However, this enzyme can be inhibited by gelatin. But on that gelatin, the structure of the gelatin is same to one fatty acid. We will talk about the fatty acid structures, but it's same to fatty acids. Goes to that side and occupy that one, not working on it. Another inhibitor. What is the inhibitor here? See, the great irreversible inhibitor, penicillin. Without knowing that, we couldn't use these things. However, finding penicillin was by chance. You know that? Yeah, that's by chance. Because they actually realized that, what is the mechanism of action, and so we know it inhibits to active site of enzyme. What enzyme? That enzyme synthesized bacterial cell membrane. So when that enzyme is inhibited, they can synthesize their cell membrane, and so bacteria have to go away. So penicillin is one of those. You wanna get familiar with this guy? Who is this? What painkiller is famous in your country? Ibuprofen is many famous in many countries. This guy was the discovered this drug effect as a competitive inhibitor. Unfortunately, he died 3 years or 5 years ago. I'm not sure when is the date of that, but yeah, it was around the
30th January
2019.
2019. Yes.
It was 95.
Where is that? Yeah. Yeah, I see the February 21. Yeah. So this is a prostaglandin pathway. You are not supposed to learn this one. Just I'm going to show you how this enzyme works and what parts they target. So here are prostaglandins. See, these are some prostaglandins. We call them P2 thromboxans. They are called prostaglandins. They cause pain and redness, inflammation after a heating, or trauma, or inflammation, everything. Prostaglandins are there. And you use some of these drugs as an anti-inflammation or pain because they cause pain and inflammation in that area. So you get some drugs like aspirins or ibuprofen. They target this enzyme. They target both same enzymes, cyclooxygenase, involved in this the downstream process. Both of them works on this, but one of them is this one is irreversible aspirin, but this ibuprofen is a competitive inhibitor or reversible. Unfortunately, when we know more, we might use them as a baby. Yeah. This enzyme, what is this enzyme? Acetylcholine esterase.
Neurotransmitter.
Yes, we have these ones in neurotransmitter pathways, and it's essential for that. When, let's say in contraction of the muscles, we release that one, and then it works on that neurotransmitter that we have there, acetylcholine there, digest that to relax that. Contraction, relaxation. So imagine you inhibit this enzyme. What happens? Contractions, and that position. Who use this kind of inhibitors? Someone or some places, they actually develop some gas agents or chemical weapons. Some of these chemical weapons, I actually have this one here. I'm not talking, I used this one, this image some years ago because at that time it was so fresh and on headlines. In the some news they actually mentioned God knows who was actually for attacking that, but some parts in the Syria use that. However, we know in some other parts of the world it has already been used in many places of the world. But yeah, one of them is sarin. What is the sarin? It's a gas agent. Actually, see, this is a nerve agent. Bonds to this active site of this acetylcholinesterase and destroy this one. When it bonds to this serine, see the OH of serine, and bonds to this one, and the bonding is irreversible and destroy this acetylcholinesterase. And unfortunately, if the levels goes up and we have a toxic level of this, it can be called main cause of this. And last part of today's session for enzyme part, what is that? Regulation of enzyme. So we know the enzymes for the drugs, but what happens in our body? How all those processes that you saw in the cells, how they are controlled? Yeah, we have several steps for controlling them. Let's then get familiar with them, some of them. What factors influence enzymatic activity? Some of them actually change enzyme level, like we produce more enzyme, but mainly, most of the time, we don't need to change the level of enzyme. Because they have activity, we change their activity. We can inhibit them or accelerate their activity. Mainly, most of the time is the best one. See, if those factors can affect the enzyme activity. What are those factors? We have even substrate. Let's say an enzyme, I'm going to work on something here. If I don't have substrate, I can go with my high speed. If I have enough of substrate, I will go in a high speed. Another thing is the what? Product. If I have more product, I don't need to work more because that product can inhibit me. We call it feedback inhibition. Feedback inhibition because we receive feedback, sometimes any product feedback inhibition, sometimes some other pathways products actually affect this one. It's not just our pathway because all of these processes are involved. And we can get some other inhibitor, like what? If the end product of this process is ATP, we have a glycolysis pathway. ATP. When the ATP is high, can inhibit this pathway. But when AMP is high, is the other forms of the energy level. AMP is a high level, AMP is a low level of energy. AMP activates this one. Let's say go. Some ATP. So it's like that. So we have a substrate and product accumulation, and sometimes when you have 10 guys in a place, you need one supervisor or one senior, sometimes 2 or 3. they need to control everything, and they say, like, you know, when you distribute something, one of them says, guys, stop that, or distribute that, or give that one. That kind of enzymes we have in pathways. In some pathways we have more than 20 enzymes, but 2 or 3, sometimes one of them are called allosteric. They regulate, they control, they actually sense everything. It seems they have several positions. They sense ATP level, they sense AMP level, they sense some controlling things. We call this enzyme allosteric enzymes. Next time if you hear that enzymes are critical enzymes, mainly they are target for the drugs. Those enzymes are important because they can control that pathway. They say go or stop. We call this enzyme what? Allosteric enzyme. Where is that? Here. Allosteric. And some enzymes are, like, let's say they are present, but we are going to change their activity so fast, activate or inactivate. That the phosphate level, some of them they get phosphate like this, activate them. Sometimes they get phosphate, they are inactivated like this. It's good. Yeah, phosphate, some other groups. It's not just phosphate. Different, but the most common is phosphate. The most common one is phosphate to control that pathway. And other one. What's the last one? This. Let's genetic control. Changing the level of them. The increased level of them. Or with hormones, we can actually change the level of the enzyme. And another one is the zymogen isoenzymes. What is this? Have you heard about this term? Yes. We have enzymes there. How happens our coagulation? When I cut a part of my body, how my enzymes start coagulating? Yeah. Exactly. Thrombin. So you have that protein, or that here are proteins. How about the digestive systems? They are same. Enzymes. We have a kind of trypsinogen converted to trypsin, or trypsinogen to trypsin. So we have a gene form of that. Those gene forms are called zymogen. They are already there, but they are not active. You just need cut part of that. With pH changes, with acting some substrate or enzyme on them, and cut part of that, that's working. It happens for fibrinogen. Fibrinogen is solid or soluble or, you know, fibrous? Soluble. Yeah, fibrin is fibrous. What happens to fibrinogen to convert to fibrin? Just a cut on protein and change it from the glucular one and soluble one to fibrous. We call this kind of proteins zymogens. They are already there. And another thing that I would like to talk about that is the isoenzymes. What are the isoenzymes? We have this? Yeah. Yes. Why? Let's say we have a liver, you know, muscles, heart muscles, brain, and all of those pathways. Do you remember that? Or the image? We have all of those processes, many of cells. So what happens? All of, you know, conditions are same in our brain, in our, you know, liver, skeletal muscles. No, they are not same. The situation is different, and the content of the substrate are different. So we need some enzyme. They catalyze same reaction. They actually do this. let's say, converting to this, bringing out or changing to that. But in some places, they should work so fast. They should work in humidity. They should work in a cold weather. Different conditions, but the same reaction. That enzymes are called IgA enzymes. They are helpful in differential diagnosis, in diseases. Yeah. For example, AST goes up, but you can find a specific AST or ALP in blood circulation because bone ALP is different from liver ALP. They are same, alkaline phosphatase. Or lactate dehydrogenase. We have lactate dehydrogenase specifically for the heart muscles, the skeletal muscles, red blood cells, and different parts of the body. So we can use that enzyme in those situations. I explained all of these things that we had in this slide. In the next slide, all of those things are related to what is allosterase, what is the feedback inhibition, final product can inhibit this pathway, covalent regulation of enzyme activity, what is this? Phosphorylation and dephosphorylation is one of them, the most common. You will see in metabolism in many places, we have a specific pathway, they phosphorylate some protein, that protein actually starts doing other things, other proteins like the dominoes actually working and starting and triggering the pathway or stopping something. We have a lot of options, so fast and in a long term. This zymogen, you see some examples here. Different zymogen, for example, carboxynogen, pepsinogen, trypsin, purine, soluble, bromine, they are already there. See, insulin is there, but as a protein soluble there. When it needs that, it's just start working that in a minute. It doesn't let you, your blood sugar goes up 400, 600 milligrams per deciliter, and your blood pressure goes up, and you're going to feel like dying. And insulin is there. He said, if I sense the glucose, I will start working. I will not let that glucose actually destroy everything. We have a lot of these things already there as a probe. So this is the definition of the IgA enzyme. They catalyze same reaction, but they are distributed in all parts of the body, and their properties is different. They have some small differences between each other. Their physical properties, or they having some extra carbohydrates, or their instability, their stability in temperature, or inhibition by different compounds, that is different. With those things, we can actually differentiate them from other IgA enzymes. And in change in enzyme abundance is related to hormones. Hormones change the level of enzymes. Last two, this image is actually from Lippincroft's book. Your reference book is Lippin. When you want to review, all things that you see here, I actually take from Lippincroft. And if you want to review those things, you can go to Lippincroft. But this one is the whole things that I explained. You can find it here. You can add your notes on this slide. Because most of the time, it gives you big picture.
So for the exam, if we study just the previous slide,
Yes, it's fine. Yeah, but I'm just trying to recommend you, if you please. However, these times, if you have any question about a specific terms, you can find in ChatGPT. Why I'm saying that ChatGPT? Because it gives you enough information about that place. And the main idea is that you get the whole concept of that. But about the details, everywhere that we have here, I mentioned that. You are not close. But for your class, I should write some more information about this, just room 18. So look, room 18 around the island. So this is my, yeah. I just did this for guys that we have in university in Tehran. When you actually go to Tehran to continue your physical part or something? Yeah, we will see you there. So my room number is 18, city center will be,
To find you or no?
yeah,
Find you or no?
yeah. And yeah, if you be in Tehran, you can find me in 18 also. Just look, every room 18, you open that. Yeah. So, so
Rest for 10 minutes.
yeah, this was for enzyme. I tried to save 1 hour, and I have saved that, but I need your help. So, I'm gonna have a short break. We're gonna have to, would you like to have a short break
Yes.
or continue that?